Thank you, Chair. My name is Rohan Baker from Genetic Signatures in Sydney, and I would like to present our validation of the EasyScreen 3base SARS-CoV-2 Detection Kit RP012 on saliva samples. And this work was primarily conducted in the laboratories of Damien Stark in the Division of Microbiology at SIDPATH in St Vincent's Hospital in Darlinghurst in Sydney. So by way of introduction, the need for accurate SARS-CoV-2 testing still remains a priority. Nasopharyngeal swabs and PCR tests are considered the most sensitive and accurate methodology, but there is a need for a less invasive sample collection method, and saliva has been investigated as an alternative sample type by many reports. However, the literature shows great variability in performance of saliva samples. So our objective was to validate a saliva sampling method on matched nasopharyngeal and saliva samples, and this was using the Genetic Signatures EasyScreen methodology already established in SIDPATH laboratories at St Vincent's Hospital. So our methodology was to collect matched nasopharyngeal and saliva samples, and we collected 104 pairs. These were collected from known or suspected COVID-19 patients in St Vincent's Hospital in Darlinghurst. The nasopharyngeal samples were collected by standard swab sampling into a VTM transport media, and saliva samples were collected using 3D printed swabs from 3D Meditech in Victoria. And there's a picture of these swabs at the bottom of the screen. The swab head itself at the top of the picture is quite large and very textured, and is designed to collect approximately 300 microliters of liquid sample. And the kit comes with a sample collection tube that contains 1 ml of liquid amines transport media. So the saliva collection method is quite critical. Saliva was collected in the morning before the patient brushed their teeth, and was collected after a deep cough or coughs with the mouth closed to bring any respiratory material into the mouth. Swab was then placed on the tongue for 30 seconds, and then followed by rotating on the tongue for an additional 30 seconds, and then agitated into the provided 1 ml of liquid amines media that comes with the swab kit. These samples then had their nucleic acid extracted using a sample processing kit, the EasyScreen SP12 sample processing kit from Genetic Signatures on the GS1 HT platform. This is shown in the center image on this slide. This is a liquid handling robot that can extract up to 96 samples at a time and then proceed to set up the PCR on those 96 eluates. So PCR detection was using the EasyScreen SARS-CoV-2 Detection Kit RP012 on a Bio-Rad CFX 384 real-time PCR instrument as shown at the bottom right. And all these samples were collected and processed at SIDPATH in St Vincent's Hospital. So Genetic Signatures has developed Threebase technology, which was used in this study. Threebase requires the conversion of the ordinary 4-base genome into a 3-base genome. And this is accomplished by a chemical modification of cytosine bases that converts them initially to uracil, and that is detected as thymine following PCR. So if we have an example sequence alignment here at the upper left of the slide, these are 10 different sequence isolates that are aligned, and there are some positions that vary in the sequence. So this region would require 48 different PCR primers to give a match to all of those 10 sequences. After 3-base conversion on the right, where all the cytosines have been converted to thymine, there is now only one position that gives variation in this alignment. And so only 3 PCR primers are required to detect all of these sequences. So 3-base can significantly reduce sequence variation in genomes, and this can lead to improved subtype similarity between closely related subtypes of a pathogen family. It also greatly simplifies PCR multiplex primer and probe design, partly by reducing the number of primers and probes that are required in a multiplex to detect multiple targets. And so this can result in improved PCR efficiency because of the more simpler nature of the multiplex PCR. An additional feature is that Threebase can bring melting temperatures of primers and probes that can be quite different based on their length and sequence composition. After Threebase conversion, it can bring those to a more similar annealing temperature, which is also important for optimizing PCR, and likewise for probes that can be quite different in their annealing temperatures. These can be much more harmonized after Threebase conversion. So importantly, the Threebase sequences will not, or assays, will not cross-react with a 4-base native sequence. And so this can reduce the potential of contamination and false positive results in the busy pathology lab. So cytosine is the base that mutates most frequently. This is random mutations now, mutates most frequently in nucleic acid. So cytosine can spontaneously deaminate to uracil. And the frequency of this is some 140-fold higher in a single-stranded molecule than a double-stranded. And as we know, SARS-CoV-2 is a single-stranded RNA virus, so it could be susceptible to this naturally occurring mutation. So as we saw from the previous slide, our Threebase technology is immune to these CT or TC mutations as well. So as a worked example of this, if we take the M gene assay from RP12, this is towards the matrix protein gene, and if we align that to all the SARS-CoV-2 sequences that were submitted to the GISAID database during July of this year, so there were 291,000 approximately sequences submitted during that time, 98% of those show a perfect match to a 4-base sequence, but there are still 2% that show a mismatch. And of that 2%, 5,700 that have a mismatch, 4,602 were due to a CT or a TC mutation. And these are the ones that are invisible after Threebase. So 81% of these naturally occurring M gene mutations are invisible after Threebase conversion. So these would be detected with equal efficiency in PCR in a 3-base assay, whereas a 4-base assay would probably have a reduced efficiency due to those mismatches under either primer or probe. So back to the results of this validation that we're presenting today. Just as a reminder, we had 104 matched nasopharyngeal and saliva pairs. So 7 of those gave negative results for both sample types. The remaining 97 were positive by at least one test. Of those, 89% were positive by both tests, 92 were positive by the nasopharyngeal test, and 94 were positive by the saliva test. So using the nasopharyngeal test as a reference, as it is the TGA validated method, this gives a positive percent agreement of 97.9% for the saliva test. If we consider all data from this study, where we had the 89 samples that were positive by both methods, and then 3 or 5 samples that were positive by one method and not the other, these samples all gave very late CT or very late CT results and are probably below the LOD of the test and thus giving sporadic positivity, and 7 negative samples. So if we consider all that data, then the sensitivity of the saliva test versus nasopharyngeal is 96.7%. And if we take all 97 positives as true positives, which we believe they are because they came from COVID-19 positive patients, the nasopharyngeal test has a positivity of just under 95%, and the saliva test a positivity just under 97%. And these positivity rates are not significantly different using a Z-score test. If we look at the cycle threshold, so the cycle threshold is where the real-time PCR curve crosses a threshold and gives a positive result in the test. We'll abbreviate that CT. If we look at the CT values plotted here in a box and whiskers plot for the 2 target genes in RP12, so N gene on the left of this image, M gene, matrix protein, on the right of the image, for both saliva and nasopharyngeal swabs. And there is no statistically significant difference in these CT results in this comparison. And the whiskers actually show that the saliva test tends to give a slightly narrower range of CT values. If we delve a little bit more deeply into the CT values from the test, here I've plotted the CT values of the N gene, nuclear protein, or M gene on the right, plotted those CT values along the x-axis, and then the y-axis has the CT delay for the saliva test. And so a negative value on the y-axis means that the saliva test gave an earlier CT than the nasopharyngeal result. So this shows that the saliva test can be up to 8 CTs earlier or later than the nasopharyngeal test, so there's quite a spread in the data range. But for both of these RP12 targets, the saliva CTs are trending earlier as the nasopharyngeal CT gets later, i.e. in samples with a lower viral load. And you can see that from the dashed blue lines of best fit that slope down towards the bottom right, indicating that on average, a saliva sample will give an earlier CT than the nasopharyngeal sample for a low viral load sample. So the saliva test may actually be more sensitive with these low viral load samples. So in conclusion, the EasyScreen SARS-CoV-2 Detection Kit RP12 has been validated on saliva samples. Saliva testing is at least as sensitive as nasopharyngeal testing when the correct saliva sampling technique is followed. And given that saliva is a less invasive sampling method, this saliva sampling works well and conveniently with the EasyScreen SARS-CoV-2 Detection Kit RP12. So this data is currently under TGA review. So finally, I'd like to acknowledge that most of this work was done in the laboratory of Damien Stark at SIDPath at St Vincent's Hospital here in Sydney for the sample testing and results. And also Dr. David Anderson, also at St Vincent's, performed some of the statistical analysis presented here today. And finally, I'd like to thank you for your attention today. And here are some contact details for myself and Damien Stark, and general contact information for genetic signatures. And I'm happy to take any questions that you may have about this presentation. Thank you.
Hi, thanks, Rohan, for the presentation. From the data that you showed, it did seem very much that the saliva test and the saliva sample collection and the nasopharyngeal collection were of similar sensitivity. That was a correct assumption?
Yes, that's exactly right, Wayne. Yep.
Yeah, right. The only question I would have, and this is not really a technical question, it's just a human nature question. Obviously, when you do a nasopharyngeal swab, or third parties doing a nasopharyngeal swab, the collection is somewhat controlled. Do you think that the saliva collection, as you described it, is open to variation more so than nasopharyngeal collection, or do you feel that that could be just as controlled?
Yeah, in this particular case, the saliva swabs were collected by a member of staff. They weren't self-collected, but certainly, yeah, I guess with any self-collection technique, if you went that way, there's going to be variability in the results, I think, from such a technique. So ideally, both collection methods would be controlled. We did try some other saliva collection methods initially, but certainly the best results we had were the ones here, where there's sort of a cough up from the lungs into a closed mouth first. Without that, the results were quite variable when comparing nasopharyngeal and saliva. We didn't extend it to something like a spitting in a tube kind of saliva collection. But yeah, as I say, these were really collected by a trained staff member for this testing presented here.
Fantastic. There are a couple of other questions in the chat line asking similar sorts of questions. Just to paraphrase them, one was, how easy was it to get a patient to cough with their mouth closed?
Well, as I said, samples were all collected at SIDPATH. I wasn't involved in the collection, so I didn't see the patients face to face. But certainly Damien Stark and his team collecting the samples didn't mention any issues with that. I think they had to give a demonstration to the patient beforehand themselves. I mean, not actually collecting a swab, but just coughing with their mouth closed. It is a bit of a strange technique, but certainly in this case, it really gave good results.
Yeah, sure. And did you do any comparisons of sample collection of the method that you described with the coughing with your mouth closed against not coughing first or collection after brushing your teeth? I mean, did you go down to that detail?
Unintentionally, yes, because the initial sample collection was quite ad hoc. It wasn't controlled at all, and we only had a very small data set, about 20 patients in that case, so I didn't present those because I didn't feel there's much power in that data set. But they were just collected any time through the day, really, and no control over the technique, and they gave often very poor results and very large CT delays for saliva. So once we researched a collection technique a little bit better, then we just went forward with that coughing sort of collection technique, and I've only presented those results because they gave a good match. The sort of ad hoc method was extremely poor, but a very small data set, so I didn't present that.
Sure, thank you. Another question was, do you have any information about how your assay performs in the detection of different variants?
Yeah, so we've certainly with RP12, we've tested that down to LOD on the major variants, alpha, beta, gamma, and delta, and they all reach the same LOD. Our assays target the nucleoprotein gene and the matrix protein gene, which are generally not mutated in any of the variants. Most of the mutation is in the spike protein. But we do undertake surveys of all sequences that are submitted, and we actually wet test any mutations that we observe under our primers and probes. And so we don't see a great effect on the assay specificity or sensitivity in that case.
Sure. And just the last question, do you have an internal control in the assay, and what is it if you do have?
We do. There's a human ribosomal RNA gene, so it just measures for load of human sample. And we have a certain C2 criteria for that to be a valid test.
Okay, well, thank you, Rohan, for your presentation and answering all of those questions. And yeah, thank you very much.
Okay, thanks, man.